All right, it's 11 o'clock, so I think we can go ahead and get started. I'd like to welcome everybody to the first seminar of the year, the first seminar of 2021. It's my pleasure to be introducing Dr. Natalie Dupre. She joined the Department of Epidemiology in 2018, but she has a long history with U of L because both of her parents taught in the chemistry department for over 30 years. She earned her bachelor's degree cum laude from Middlebury College in Vermont as a neuroscience major and a Spanish minor. Her master's and doctoral degrees come from Harvard T.H. Chan School of Public Health in Boston, where she was a David H. Piper's Fellow. Then at Brigham and Women's College in Boston, she was a postdoctoral research fellow in cancer epidemiology. Dr. Dupre's expertise encompasses cancer epidemiology, environmental epidemiology, and molecular epidemiology. Some of the exciting research findings she has published include the role of particulate matter exposure on worse breast cancer survival and the potential role of radon exposure influencing estrogen receptor negative breast cancer. I'm excited for this presentation because Dr. Dupre is not only an educator and a researcher, but she is also an excellent science communicator, which I found out by listening to her podcast, Pandemia. So welcome, Dr. Dupre.
Thank you, Jennifer, and thanks to the organizers for getting this together and inviting me to talk to you all today. I'm going to attempt to share my screen and get into presentation mode. Can you all see can people see my screen okay?
Yes, we can.
Okay, thank you so much. And I'm not able to view the Teams interface, so if there are hands that pop up or comments that pop up, you'll have to let me know throughout the presentation.
Dr. Dupre, just a quick suggestion, if you could switch your pointer to a laser pointer, if you right click on it, you could be able to change that. That'll help us see where you're pointing.
Okay, is that helpful?
I think so. Thank you.
Okay, great. Well, today, I must admit, it feels a little strange to be giving a presentation today after what's happened in our country, but I will go on to talk to you all about the research that I've been a part of and am helping to start up here in Kentucky. Related to environmental exposures to air pollutants and radon and how they influence breast cancer development. Just to give you a little bit of an overview of my talk, I'll be giving some background about breast cancer epidemiology and then going into the work that's been done on radon and breast cancer, followed by work that I've led on air pollutants and breast cancer. So just to give you all an idea, I'm sure you know that the burden of breast cancer is very large. It's the most common cancer diagnosed in women, excluding non-melanoma skin cancers. Here you can see a map from the WHO's International Agency for Research on Cancer showing age-standardized breast cancer rates across the world. And you can see the areas in dark blue are areas of high breast cancer diagnoses, and these are predominantly driven by the high screening rates in these countries. We have screening through mammography. And just to give you some concrete numbers, in the United States, about 250,000 women are diagnosed with breast cancer each year. And in the United States alone, around 40,000 women die from breast cancer each year. One of the big advancements in breast cancer epidemiology has been the identification of breast tumor subtypes through molecular characterization of various hormone receptors, namely the estrogen receptor and progesterone receptor. This really has changed the game in terms of breast cancer treatment, where now for women whose tumors contain estrogen receptors, hormone treatments have been very successful. and there's another receptor called the HER2 receptor that can be treated with the Herceptin treatment and has been helped a lot in terms of treating breast cancer and improving breast cancer survival rates. Here I've listed out the 5-year breast cancer survival rates for the different tumor subtypes, where there's over 90% of cases with hormone receptor positive cancers will survive 5 years. However, the breast cancers that do not exhibit hormone receptors, these are here at the bottom, hormone receptor negative and HER2 negative, the 5-year survival rate is only 77% due to the lack of treatments for estrogen receptor and PR receptor negative tumors. One thing that's really important about this is that there are racial differences in the distributions of hormone receptor breast cancers. Here is some data from the American Cancer Society's Breast Cancer Facts and Figures showing the distribution of breast tumor subtypes by different racial and ethnic groups, where you can see in the second column here, we have the non-Hispanic Black population, and these gray areas show the proportion of breast tumors that are hormone receptor negative and HER2 negative. Those are also called triple negative breast cancers. So you can see about 21% of the cancers in non-Hispanic Black women are these more aggressive estrogen receptor triple negative breast cancers, whereas in non-Hispanic white women, that's around 10%. So the big point here is that these we need to figure out ways to identify risk factors for triple negative breast cancers, because we don't have very as effective treatments for estrogen receptor negative breast cancers. I think it's also important to point out some important literature on the role of policies that may be influencing these disparities in estrogen receptor negative breast cancer. This work, led by Dr. Nancy Krieger from the Harvard School of Public Health, was published in 2017, looking at the role of Jim Crow laws in estrogen receptor negative breast cancer rates across the United States from 1992 through 2012. And what they did was they looked at the SEER cancer registry data from 13 states and gathered data from all the primary breast cancer cases that occurred between 1992 to 2012 for women ages 25 to 84 years old. So in this analysis, they included around 47,000 cases of cancer for Black women and around 350,000 cases of breast cancer from white non-Hispanic women. And what they did was they categorized whether or not the cases were born in a state that had a Jim Crow law. The Jim Crow laws were essentially legal forms of racial discrimination. In addition, and you can see this map on the right-hand side, the states in red in Washington, D.C. Were the areas that had a state or local Jim Crow law. In addition, they obtained potentially important covariate data on age, year of birth, tumor characteristics, and neighborhood SES to adjust for potential differences in tumor characteristics and SES. And what they found here was that Jim Crow laws affected estrogen receptor negative breast cancer rates. They had 3 major findings. One, that being born Black in a Jim Crow state was associated with greater odds of developing estrogen receptor negative breast cancer as compared to being born Black in non-Jim Crow states. You can see that adjusted odds ratio is around 1.1, and the 95% confidence intervals are all above 1. However, being born white in a Jim Crow state was not associated with greater odds of estrogen receptor negative breast cancer as compared to being born white in a non-Jim Crow state. So this highlights the inequity of being Black in a Jim Crow state. Additionally, another major finding was that the racial inequities, comparing Black versus white women in terms of estrogen receptor negative breast cancer, was stronger if the case was born during Jim Crow prior to 1964. So being born during the Jim Crow era, the odds ratio of estrogen receptor negative breast cancer comparing Black women to white women was around 1.4, and this was attenuated for the cases that were born after Jim Crow laws, where the odds ratio was attenuated down to around 1.3. So there's still racial inequities even after Jim Crow, but it was much stronger during Jim Crow. And I think this is important to highlight because there are racial disparities in estrogen receptor negative breast cancer, and estrogen receptor negative breast cancer is the cancer that is has less treatment and is more aggressive in terms of causing having worse breast cancer survival. The real question is what is driving these disparities? And for that, I am going to focus the rest of my talk on in the environment. And people, some of the first studies that came out highlighting the role of the environment in breast cancer were migration studies that happened during the 60s when, and this was also the time when state registries in the United States were starting to collect cancer development in states. So what this study was conducted or published in 1973, they found that the, let's see, so the black line here shows breast cancer rates in Japan, and the orange lines here show the rates of breast cancer for Japanese women who immigrated to Hawaii and San Francisco. And the red line shows breast cancer rates in white women in San Francisco. And as you can see, that the Japanese women who immigrated to the United States start to take on the breast cancer rates of women in the United States. So this suggests the role of the environment, as genetics are not changing for these first generation Japanese Americans. And so there's been a lot of research on what could be some of these risk factors that immigrants may take on when they come to a new country. And I think we've done a lot of work to understand breast cancer risk factors. I'm not going to go through all of these breast cancer risk factors, but I think it's important to know that there are very few modifiable risk factors listed here. So we have reproductive factors, so whether or not a woman has a child, the age at which they have the child, whether or not they can breastfeed. The other modifiable lifestyle factors include BMI and physical activity and alcohol consumption. So there's still a lot of room for the role of the environment. And in the early I think it was 2013, the Institute of Medicine called on researchers to study the role of environmental factors in breast cancer. And it was a joint, there was a joint effort by the NCI and the NIEHS to start to fund research at the interface of environmental factors and breast cancer. So that, honestly, I got into this because out of that collaboration, my mentor received funding for a grant to study the environment and breast cancer, and that was what my dissertation work and postdoctoral work was on. So anyways, it's relatively new to be studying the environment as I am discussing it. But one important thing, finding that I'm going to highlight is that high-dose ionizing radiation exposure, particularly if exposed in early life, is a well-established breast cancer risk factor. So there have been two pivotal studies on high-dose radiation exposure. So the lifespan study was looking at women who had survived the atomic bomb in Japan and looked at, followed them for development of breast cancer. And this figure here is showing the excess risk of breast cancer for a unit increase in radiation exposure from the bomb. And if the woman was, at the time, a girl, less than 10 years old, the risk of breast cancer was much higher than if the woman was exposed to the atomic bomb later in life, around age 35. So there's still an elevated risk, much higher, very high odds, relative risks here if they are exposed to high-dose ionizing radiation. Similarly, a study conducted on tuberculosis treatment, and this was where, this study was done in Canada, where there was national treatment of tuberculosis, and they were giving repeated chest x-rays to women for treatment of tuberculosis. And they followed these women and observed that women who were repeatedly exposed to fluoroscopy early in life, at ages 10 to 14 and 15 to 24, that their relative risk of developing breast cancer later in life was 4 times higher for a unit increase in radiation exposure. So this all ties into, we can think of the biologic mechanisms that are in play. Radiation's obviously going to cause severe DNA damage, and that it might be leading to the breast cancer development. So clearly, high-dose ionizing radiation exposure is a breast cancer risk factor, and these are just two of the most pivotal studies about that. But we often, there's been much less literature out there on radon exposure, a naturally occurring form of ionizing radiation that people are exposed to predominantly in their homes. So if there are cracks in the foundation of your home, radon can enter in through the soil and into the home, and you can be exposed to radon that way. There's ways to remediate radon through radon remediation, and actually the state of Kentucky, you can get a free radon test kit if you go to the Kentucky State's radon program to measure radon in your own home. So I became interested in this during my postdoctoral research, and worked with a great team of researchers to look at the role of radon in development of breast cancer. The cohorts that we worked with were the Nurses' Health Study and the Nurses' Health Study 2 population. So just to give you an idea of these cohorts, they're prospective cohorts. The Nurses' Health Study started in 1976 and enrolled 121,700 female nurses who were at the time ages 30 to 55 years old. And every 2 years, these women are mailed a questionnaire to ask them about any new diagnoses that they've had, their medical history, reproductive factors like the number of children they've had and when, various lifestyle factors like physical activity and diet, and we've also asked things about health behaviors. So, vitamin use, medication use, it's a really rich database. And so the Nurses' Health Study has sent out questionnaires up until 2018, now that the nurses are entering old age and starting to develop cognitive decline. But the Nurses' Health Study 2 started a bit later in 1989, enrolling 116,430 female nurses that were younger, ages 25 to 42 at the time. It follows the same model where every 2 years the participants are mailed questionnaires to ask about their diagnoses, medical history, lifestyle factors, and so on. One important thing about these two studies is that they cover a large, they cover every single state in the United States. I'm not showing Alaska and Hawaii here, but these maps here, each dot represents the residential address of these hundreds of thousands of participants. And so you can see that there's quite a great coverage across the United States. And this enables us to look, really examine environmental exposures because we have such great exposure variability. So this work was led by a colleague, Trang Vo Pham, and she did a radon exposure model, a collaboration with the Lawrence Berkeley National Laboratory that estimated indoor radon concentrations for every county in the contiguous US. And here's a map on the right of radon concentrations throughout the United States. Yeah, you can see, I'll show a map a little bit later, but even within Kentucky, there's quite a bit of variability where those areas in red are the areas of high radon exposure that are above the EPA limit of 4 picocuries per liter. But the cool thing is with our Nurses Health Study participants, we can link the participant's residential address to measure these estimated measures of radon. So if I live in Kentucky, I will pull out, if I live in Louisville, I would have Jefferson County's radon concentration. So the results from this analysis found that women who lived in these are adjusted hazard ratios. We adjusted for multiple breast cancer risk factors, and the dotted line here on one is the null value, and you can see that women who lived in the highest quintile of radon exposure, county level radon exposure, were about 1.4 times more likely to develop estrogen receptor negative breast cancer. That's indicated, and that p-value for trend was significant at the 0.05 level. And as we further defined the breast cancer outcome by not only estrogen receptor, but also progesterone receptor, we can see that consistent nearly dose-response relationship in terms of quintiles remained. However, we lose a few cases. And then when we further look at triple negative breast cancer, that's ER, estrogen receptor, progesterone receptor, and HER2 negative, the association remains even though we only have about 293 cases. So as our outcome, our breast cancer outcome, gets more and more well-defined, the association between high radon exposure remains for these triple negative breast cancer. In comparison, radon was not associated with estrogen receptor positive breast cancer. So this was looking at county level radon exposure. So we weren't really sure if there was how much measurement error. But one additional thing that we did, and this brings in the molecular epidemiology piece, is that for the participants in the Nurses Health Study and the Nurses Health Study 2, we collected archival breast tissue from these women. So if the woman underwent a lumpectomy or a mastectomy, we asked the hospitals for that tissue in formalin-fixed, paraffin-embedded FFPE case. This is what they look like here. So what we did was we took cores from the tumor area and cores from the adjacent normal area, and we measured RNA transcripts to measure breast tissue gene expression in both the tumor and the adjacent normal areas. So we have about 18,000 genes that we measured expression for, and we had around 874 tumor samples and of fewer normal samples, around 700. What we found here, and I'm sure there's a lot of more basic scientists that could understand this better than me, but what we observed was that radon was associated with upregulation of genes involved in MAPK signaling, as well as downregulation of the phosphocholine synthesis pathway. And these are, so here we've got the MAPK signaling pathway and the genes that were measured and involved and contributed to the pathway the most. And yeah, I will leave it at that for the sake of time, because I'm really excited about some of the work that we're going to be starting up this year. So these studies were some of the studies that I showed, are some of the first studies of prospective cohorts. However, it's limited because we had used county level radon exposure. So we don't actually know the individual radon exposure in the home. So there are currently no studies of personal exposure to radon in development of breast cancer. So we recently received funding from U of L to measure residential radon concentrations in 100 breast cancer patients at the U of L James Graham Brown Cancer Center. And so we're going to get these free radon kits that are also used by the state, and we're going to send them out to enrolled participants and ask them to return the radon kits so that we can measure them. Part of it's to figure out whether or not we can actually use these kits in epidemiologic studies. And the figure on the right kind of shows the distributions of radon within Jefferson County. This is thanks to Connie Mendel at the Louisville Metro Department of Public Health and Wellness. And you can see the eastern and southern parts of the county have the highest radon levels. But there's also quite a bit of variation within zip codes that's not described by this map. We are also starting a project to look at Kentucky breast cancer cases. We recently received the KCR, which is the Kentucky Cancer Registry data for breast cancer cases, and we are hoping we are going to be working to link the geocoded addresses of breast cancer cases in Kentucky from 1995 through 2018 and link them to county level radon levels. So I'm really excited about working with this data set, and hopefully we'll have some results to present by the end of the year. But here on the right-hand side, I just wanted to show the distribution of estrogen receptor, PR receptor, and HER2 receptor breast cancer across the state. You can see that there is quite a bit of variability across the state in terms of triple negative breast cancer rates. So I think this will be interesting, and I hope to share some results at a later date. I'm now going to move into talking about air pollutants and breast health within the Nurses Health Study and some of the projects that we are starting now. I'll focus on particulate matter and hazardous air pollutants. Sure, members know that particulate matter is a group 1 human carcinogen, meaning that it is carcinogenic to humans. Particulate matter is PM 2.5 is particulate matter that's less than 2.5 microns, and its predominant source is through combustion of particles. They're produced through combustion sources, things like traffic exposures, and we also have PM 10 that's larger in size, less than 10 microns, and the predominant sources are mainly through dust and anthropogenic sources like pollen and mold. So the work that has been done within the particulate matter is an EPA criteria pollutant, so it's required to be measured through the Clean Air Act. And here's a map on the left of the monitors for particulate matter. What the Nurses' Health Study has done is estimated particulate matter exposures across the United States. So here's an example on the right showing PM 2.5 distributions across the United States. And so we have these spatiotemporal models to estimate monthly exposure to PM 2.5, the larger coarse size fractions from 1988 through 2007. So here's an example of 1 month in June 1989. And again, we are able to link the participant's address to these estimates of PM over time. Additionally, the US EPA is required to regulate air toxics or hazardous air pollutants, also because of the Clean Air Act. So we looked at the US EPA's assessment of air toxics. This is through NATA, the National Air Toxics Assessment, and they examine things like benzene, dioxins, metals, toluenes, and much more. In 2002, they measured 181 hazardous air pollutants through emissions data and doing dispersion modeling to account for things like meteorology that influences the transport of these pollutants. Many of these hazardous air pollutants are considered mammary carcinogens and endocrine disruptors. So this work was led by Dr. Jamie Hart at Harvard and Brigham and Women's, and we observed that particulate matter exposure was not associated with breast cancer risk, regardless of estrogen receptor status. So here again are the adjusted hazard ratios, and for a 5-unit increase in PM, you can see that the 95% confidence intervals cross the null value of 1, meaning that the results are consistent with no association. And the blue represents PM 2.5, the gray is PM 2.5 to 10, and the green is the larger size fractions of PM 10. And these were adjusted for many different breast cancer risk factors. This finding of no association between PM 2.5 and breast cancer incidence has been observed in multiple other cohorts, including the sister study, the Danish nurse cohort, and a meta-analysis of European cities. So I think it's pretty well understood that PM 2.5 is not associated with breast cancer incidence, and I'm realizing that this is not a totally complete list. There have been a few more studies on PM 2.5 and breast cancer incidence also showing no association. But the story is a little bit different between particulate matter exposure in relation to breast cancer survival. And I think it has to do with the role, the biological role of PM in the body. So particulate matter promotes systemic inflammation, oxidative stress, and epigenetic changes, all of which are relevant for breast cancer survival. In the epidemiologic literature, inflammation-related exposures are much more strongly associated with breast cancer survival as opposed to breast cancer incidence. So, for example, aspirin is an anti-inflammatory drug, and it has been strongly associated with better breast cancer survival, but has not been associated with breast cancer risk. So that for those that consume aspirin regularly, the hazard ratio was I think it was around 0.4, and that work is done by, led by Dr. Michelle Holmes. So I was interested in whether particulate matter influenced breast cancer survival, and this was the focus of my dissertation, and I'll show you all a little bit about the findings there. So within the Nurses' Health Study and the Nurses' Health Study 2, there were around 9,000 women who were diagnosed with stage 1 to stage 3 breast cancer between 1988 through 2007, which are the years for which we have particulate matter data. And we gather information from medical records that we've requested from their doctors on stage, estrogen receptor status, grade, and treatments. In addition to all of the questionnaire data that we have about lifestyle factors. And so our exposure for this analysis was particulate matter exposure that was updated every 2 years, and this was post-diagnosis particulate matter exposure. And over the course of the follow-up of these 9,000 women or so, around 1,200 women died from breast cancer. And what we observed was that when we looked at all of the 9,000 women diagnosed with breast cancer, it didn't appear that particulate matter was associated with breast cancer survival. These hazard ratios and the 95% confidence intervals are very close to the null value of 1. But when we looked by stage, which has been previously reported as an effective modifier in studies of PM and breast cancer survival, we observed that for a 10-unit increase in PM 2.5, the hazard ratio was around 1.6, and this was significant, whereas the larger size fractions did not appear to be associated with breast cancer survival among stage 1 women. When we looked at more advanced stage women with more advanced stage diseases, it did not appear that particulate matter was associated with breast cancer survival. And these were adjusted for various predictors of mortality. So there have been 4 other studies looking at PM 2.5 and breast cancer survival, and it's actually quite consistent across the literature, showing that PM 2.5 is associated with higher breast cancer mortality, particularly in stage 1 women when that was looked at. So the first study on this topic was done with the California SEER data, showing that for a 5-unit increase in PM 2.5, the hazard ratio was 2 times higher for a 5-unit increase in PM 2.5. A study in Italy that did not stratify by stage also was looking at pretty much a 5-unit increase and saw an association. However, these the Italian study and the SEER-based studies were not able to account for as many confounders as we were in the Nurses' Health Study. Since a more recent study came out that looked at all of the US SEER data, and again is showing a suggestive increase in breast cancer mortality for a 10-unit increase in PM 2.5, but again, with the SEER data, we're limited by what can be adjusted for. In terms of the hazardous air pollutants, there have been studies done in the Nurses' Health Study 2 that I'm going to present, and there's also been 2 studies published within the sister study cohort that's also a nationwide cohort. It's pretty hard to study these because these hazardous air pollutants are measured at the census tract level. And again, we're using the NADA data for this. That's not conducted every year. But there were some interesting suggestions of associations between specific hazardous air pollutants and breast cancer incidence within the Nurses' Health Study. I won't list out all these names, but here were a few of the hazardous air pollutants that were suggestively associated with higher risk of estrogen receptor positive breast cancer and estrogen receptor negative breast cancer on the right. So I recently received funding from U of L to enroll 100 patients. Sorry, just one more thing. The studies on hazardous air pollutants and breast cancer incidence was again looking at census tract level concentrations of hazardous air pollutants and not actually measuring an individual's exposure to these air toxics. So what I'm hoping to get started with this year is the study that would fund the measurement of over 1,500 chemicals, environmental chemicals, including some of the air toxics that were in the previous publications. So we're planning to enroll 100 patients with estrogen receptor positive and estrogen receptor negative invasive breast cancer from U of L's James Graham Brown Cancer Center. And we will ask them to complete a questionnaire on breast cancer risk factors that models the questionnaire questions from the Nurses' Health Studies. But the key contribution of this grant funding will be to examine the feasibility of wearing these myexposome silicone bracelets to measure over 1,500 environmental chemicals. And this includes phthalates, benzenes, BPA, polycyclic aromatic hydrocarbons, PCBs, dioxins, volatile organic compounds, and pesticides. So we have funding to measure 20 participants' environmental chemical levels. They're quite expensive, these silicone wristbands. They're about $1,000 to measure 1,500 and more chemicals. And it requires that the participants wear the bracelets for 5 to 7 days to be able to adequately detect these environmental chemicals. And of course, it's not only air toxics, but it's also measuring skin toxics, and we won't be able, or skin dermal exposures. So we won't be able to tease apart dermal versus ambient. But I think this is a good step to see what people are exposed to in the Louisville area and figure out if there are any differences in exposure levels between ER positive and ER negative women. So it's obviously not a perfect study because we don't have controls or people without breast cancer, but this will be good pilot data that I'm hoping will be important to include in a larger grant to measure environmental exposures in cases that are documented through the Kentucky Cancer Registry, and also in controls or women without breast cancer. Additionally, hopefully, again, with the Kentucky Cancer Registry, we've obtained the geocoded addresses of all breast cancer cases that have been identified in Kentucky since 1995. And we can link those geocoded addresses to estimates of hazardous air pollutants and again, compare levels of hazardous air pollutants in women with ER positive disease versus ER negative disease. And again, this will be an ecologic analysis, but hopefully if the myexposome wristbands work, we could potentially scale this up and ask women who've been diagnosed with breast cancer to wear these bracelets to conduct a breast cancer case control study. So I'd like to acknowledge the Nurses' Health Study and the Nurses' Health Study 2 participants and staff who have been working tirelessly since the 70s. An entire issue of the American Journal of Public Health has been devoted to the contributions of the Nurses' Health Study participants and all the research that has come out of that cohort. I'd also like to thank my collaborators at U of L School of Public Health that are and U of L who have really made it possible to start studies of environmental exposures in breast cancer. And I'd also like to thank my my colleagues from the Harvard Chan School and Boston University and UC Irvine. And importantly, I'd like to thank the research funding support from the center, U of L's Center for Integrative Environmental Health Sciences, some funding that we got from the School of Medicine's Jewish Heritage Fund for Excellence, and U of L's EVPRI that will be funding the myexposome and radon kits, as well as support from the training grant that funded me throughout my postdoc and doctoral program. And the Susan G. Komen Foundation that funded the work on particulate matter exposure and radon exposure, and these larger grants that funded the development of the Nurses Health Study cohorts and the breast tissue gene expression collection. So with that, I'll end my talk, and thank you for your attention. I hope that I know that went pretty quickly, but hopefully it was you were able to understand, but I'm happy to take questions.
Thank you, Dr. Dupre, for the interesting talk. It was really thought-provoking. I'll invite people to either put your questions in the chat or raise your hand. And Dr. Huntington Moscos had a comment in the chat. She says, I am in the Nurses Health Study 3, and her mother-in-law is in the cohort 2. So it's very well known among nurses.
And the Nurses Health Study 3 is quite different because they're recruiting through by using electronic communication. So instead of mailing questionnaires, they're now contacting these Nurses Health Study 3 participants via email and phones, and they're actually very much focused on environmental exposures. So the Nurses Health Study 3 cohort will be an incredible resource for understanding environmental exposures and health. So thanks, Liz, for contributing. That's incredible.
All right. Johnny Wise has a question.
Natalie, great presentation. I really enjoyed it. I was wondering, so cancer, of course, takes years to decades for onset, and Americans have a habit of moving a lot. Mm-hmm. So I was wondering how you factor that into your radon exposures and cancer outcome.
So because we're mailing questionnaires to the Nurses Health Study participants every 2 years, we get updated addresses every 2 years. So if, let's say in 1988, I lived in Jefferson County, I would get the radon level in Jefferson County in 1988. But if I moved to California in 1990, then I would be assigned the radon level in that California county. So it gets updated, the radon levels get updated as the person moves. And it's all because we are mailing these questionnaires and have those updated. But you know, it's not perfect in that, you know, we're only doing it every 2 years. So if a woman moves, you know, after 6 months, we're misassigning the exposure for a year and a half, essentially. So it's again, there is measurement error in the exposure levels. And I think that's kind of also an interesting thing. You would expect the measurement error to not be differential in terms of cases and non-cases, which would bias the results toward the null. So what we're observing with radon and estrogen receptor negative breast cancer is, under that logic, is actually an underestimate in terms of if we had measured it, if we hadn't mismeasured radon exposure.
Thank you for that. Dr. Hong has a question.
Yes, thank you for a wonderful presentation. There's so many interesting implications from the studies you presented. My question is kind of related to what Johnny just talked about, because you mentioned that age matters. You know, that when the exposure, you know, ionizing radiation in terms of, you know, early on, you know, kids are more susceptible. And also there's a racial part in that. So I was thinking so I guess it, you know, wouldn't it matter more, you know, to figure out where they grew up rather than where they're living now, in terms of looking at these data, first of all. And then I was wondering if you also found a similar racial inequality in the nurse study cohorts.
Okay, so for the first question about, or basically, so I'm hoping, at least for the studies that we're going to conduct here within Louisville, through that pilot study, I forgot to mention that I'm going to ask about residential histories during these important time windows like puberty, when the breast tissue is changing dramatically and appears to be most vulnerable to environmental factors. But I do think that, that's one area that's a huge gap right now in terms of breast cancer epidemiology in general, that we do need to be asking what is happening in adolescence and early adulthood. Another important time window of exposure is when a woman is pregnant and lactating. That's another vulnerable moment for the breast tissue. So yeah, I think that's all super interesting. And by asking residential histories, we're starting to get at it. I think it would be really awesome if we could start a cohort of, teenage girls and follow them. But the feasibility of all of that is really challenging, and I haven't wrapped my brain around that enough to implement things. As far as the racial differences, so within the Nurses' Health Study, it's predominantly non-Hispanic white women. Within the Nurses' Health Study 1, that don't quote me, but it's over 90% of the participants are white. The Nurses' Health Study 2 is a bit more racially diverse, but still like 85 or so percent white. So we didn't really have enough power to separate out by race. But there is a lot of literature that Black, non-Hispanic Black women have stronger associations with other health outcomes. So I would expect that if we were to do that, we might see stronger associations among non-Hispanic Black women, but we just weren't powered enough to really answer.
That's interesting, because it seems to me that racial inequality itself is affecting investigators more, well, I guess, unable to study racial inequality, because all the nurses being mostly, white and things like that. Anyway, fascinating.
There is another cohort that might be able to answer this, the Black Women's Health Study, but they have not reported yet on any of these environmental exposures yet. But that would be a great cohort to follow up with. There's also there's other cohorts that could do that if they have researchers interested in environmental epi. And again, this is kind of like relatively new, like the last 15 years, the literature on environmental exposures and breast cancer has been exploding really from essentially nothing. So there's a lot of, good points.
Thank you.
Thank you. Dr. Lukai has his hand up.
Oh yeah, this is an excellent talk and bring a lot of interest. And because two other person already question about the model, so I also interesting about the model. Like you are mentioned about the Japanese Tom bomb survivor, and they have kind of the age-dependent and breast cancer interest. But that is a different condition. That is the predominant got is external exposure, probably the gamma radiation or X-ray radiation, and radon, and that is should be predominant in healing and the inhibition to the lung. So most times radon induced lung cancer, and so in that case, so I don't know what your rationale is, and inhalation radon and cause breast cancer, and for your one study, and you have see the trend and kind of increase, but not that like a big variation. And you're in the fake legend, and I read you're already limited a lot of the confounders, but for example, smoke or something like that. But I didn't see you have think about the heavy metal, because like a couple of the heavy metal have some gender difference, like cadmium, that is kind of estrogen kind of like compound, and that sometimes affect the estrogen induced cancer. And the cadmium also induced breast cancer, actually. So probably that is the one confounder when you measure the sample need to think about. And the dose, and if you are measuring the basement or the house, that is the air concentration, and you need and check with the patient how long they will stay in that room, and because that is inhalation is more important. So that is what I'm thinking. Thank you.
Thank you.
Dr. Kwokon, do you have a comment or question?
Yes, Jenny. I know that we have time for everybody to go somewhere else, but thank you, Natalie, for the nice presentation. One part of my question actually was about looking at other type of cancer, like Dr. Kai just mentioned. But the other part is very naive question. What do you do when you find out about radon exposure in your home? Do you just have to move, or what do you do in that case?
Oh, okay. Yeah. So the nice thing about radon is that there are pretty straightforward interventions to remove radon from the home. Radon remediation. It's not necessarily inexpensive, but it's about somewhere around $1,000 to remediate one's home from radon. And basically it's a ventilation system that there's a pipe that goes on the outside of the home to release radon from the home out above the roof of the home. So that's, so that it's a pretty simple, relatively simple. I think it's probably easier to do radon remediation than try to get someone to quit smoking. But yeah, so that's how one gets rid of radon in the home. And that is one of the questions. Many homes do have that system already in place, particularly if it was purchased the home was purchased more recently. Yeah, and so just to kind of follow up on the confounder discussion, I wasn't exactly catching every single confounder that might have been listed by Dr. Kai, but so cadmium, I think, was raised as one. So we did adjust for smoking, and it didn't make a difference, but we just don't have cadmium levels measured in the participants. It's also not necessarily from what I'm aware of in terms of the epidemiologic literature on cadmium and breast cancer, it's not a strong breast cancer risk factor. So I doubt it would, by adjusting for cadmium, that the results would change that much. But I do think it's interesting to look at. And yeah, so we've adjusted for smoking as a surrogate for many things, but one of them might be cadmium.
Thank you.
Thank you. We just have a couple of minutes left, so I'd like to remind those in the Cancer RIG that we have a meeting right after this in another Teams link. And I had one last question for Dr. Dupre, kind of related to the radon in the homes. And if the levels that you showed in it countywide or in Jefferson County in particular are the levels that are detected in homes, or if those are the levels in the environment, and how much radon is related to the maintenance and health of the home. The racial disparities that you talked about kind of made me realize that those Jim Crow laws, one of the discrepancies would be in housing opportunities, whereas in the Jefferson County map that you showed me, the high levels in the east I associated with, nice neighborhoods.
Yeah. So that map that I was showing you are for people in Jefferson County. It's residential at the zip code level. So these are people who have requested from the state or the Louisville Metro to have radon measured. So, and if you, it was hard to notice, but on the western side, a lot of the counties had very few number of homes do the radon sampling. So let me just try to figure out how I was going to say this. But yeah, so these are people who are, concerned about radon that have it measured. It's by no means represented, even within a zip code, it's not representative of the entire zip code. Yeah, I think the work that we're doing, that we're going to try to do at the state level, will really help us to understand, hopefully, these racial disparities. But we need to do the studies, to answer that second part of your question.
Thank you so much. A lot of discussion on this, and it was a very great talk. So thank you, everybody, for being here.
Thank you for your time and attention.
Thank you.